Welcome to A2 Genetic Technology. In your AS level course, you explored the natural structure of DNA. Now you are moving from merely observing biology to actively engineering it. This is the applied science driving modern medicine, agriculture, and forensics, and a major component of your Paper 4 exam. This unit is all about the precise molecular toolkit scientists use to isolate, manipulate, and transfer genetic material between entirely different species. We will look at how we manage the sheer volume of genetic data and bridge the gap between laboratory techniques and real-world applications. We begin with the absolute foundation, recombinant DNA technology. You will master the biochemical tools required to cut and paste the genome. First, you will meet restriction endonucleases. These are the molecular scissors that hydrolyze phosphodiester bonds at specific recognition sites, leaving staggered sticky ends. Then, you will see how DNA ligase acts as the essential molecular glue, sealing the sugar phosphate backbone to permanently join fragments from different organisms. Once you have cut your target gene, how do you deliver it into a new cell? You will explore the anatomy of vectors, such as plasmids, and learn why features like marker genes and promoters are critical. We then look at gene cloning, where we insert these recombinant vectors into host bacteria, using them as living photocopiers to multiply the foreign gene on an industrial scale. But what if you need to amplify DNA without using living cells? You will learn the exact temperature cycles of the polymerase chain reaction, or PCR, which uses Taq polymerase to exponentially amplify microscopic DNA samples in hours. Once amplified, we use gel electrophoresis, a technique that applies an electric field to sort invisible DNA fragments by their size. Next, we move to reading, analyzing, and editing the genetic code. You will uncover how DNA sequencing determines the exact order of nucleotides and how bioinformatics uses computational databases like BLAST to compare those sequences across species. We also dive into CRISPR-Cas9, a revolutionary system using guide RNA to target specific sequences, allowing scientists to edit genes with unprecedented precision. All this molecular theory builds to large-scale genetic engineering and real-world applications. We will investigate how genetically modified organisms are created to improve crop yields, like engineering herbicide resistance in agriculture. You will also see how industrial microbes are genetically engineered to produce vital human proteins, such as insulin, transforming modern medicine. Because the power to alter the genome is so profound, you must also learn to navigate the medical consequences and ethical responsibilities. In human medicine, we will explore gene therapy. You will learn how viral vectors are used to treat severe genetic diseases like SCID by directly replacing defective alleles in a patient's cells. Finally, you will rigorously evaluate the ethical, social, and environmental implications of genetic technology, constructing balanced, mark scheme-compliant arguments for your Paper 4 exam. By the end of this unit, you will have mastered the molecular toolkit of genetic engineering, and you will be ready to tackle complex Paper 4 data questions.